Thank you. Good afternoon, everyone. It is great to be back here at ARM in person. Frankly, to be out of my slippers and basement office is very nice, although I do miss my slippers. I'll skip over the forward-looking statements. So Amicus Therapeutics is a fully integrated rare disease biotech company. The foundation of Amicus is Galafold, which is our small molecule chaperone precision medicine that's marketed for Fabre, where we have a commercial footprint in over 27 countries worldwide. The next big step for Amicus is ATGA. That is our next generation ERT that's co-administered with a small molecule stabilizer that is currently in registration for Pompe disease. And for the future, Amicus has a growing portfolio of gene therapies, including 2 clinical programs in Batten disease and a discovery engine that combines Amicus protein engineering expertise and platforms with University of Pennsylvania expertise and platforms in AAV vector technology. So the 2 lead programs for Amicus and Fabre and Pompe both have a potential for $500 million up to $1 billion in peak sales. First, we have Galafold, shown here on the left, where the launch is progressing nicely. Galafold was first approved back in 2016, and this year we're on track for between $300 and $350 million in revenue. Galafold is projected over the next couple of years to reach $500 million. Second, on the right, we have ATGA, where we're currently undergoing submissions in Pompe disease. We're on track to kick off the launch next year, and recently announced FDA accepted the BLA and NDA for ATGA. And we're really excited for the potential to continue ramping up access to Galafold and then hopefully for ATGA worldwide to improve lives of patients and families living with Fabre and Pompe. So about 2 weeks ago, we were really excited to announce that we are spinning out our gene therapy business, combining it with REF4 to launch Castelli Therapeutics, which will be a new independent publicly traded company with about $400 million in initial funding. This separation will sharpen the strategic focus, financial profile, and operational execution for both companies. And this will transform Amicus into a late-stage development and global commercial rare disease company and accelerate its path to profitability, which we now anticipate in 2023. So importantly for Amicus, we will remain a mission-driven, patient-dedicated rare disease company. The transformation allows Amicus to grow its leadership position in Fabre and Pompe disease. In Fabre, Amicus will continue and accelerate our geographic and label expansion for Galafold, and importantly now we can support new diagnostic initiatives given how underdiagnosed Fabre remains today. For Pompe disease, Amicus will continue to focus on securing global approvals and executing on the global launch of ATGA. Importantly, Amicus will continue developing Fabre and Pompe gene therapies together with Keratos. There will be a 50-50 co-development and profit-sharing collaboration between the 2 companies. And additionally, Amicus will have rights of negotiation to future muscular dystrophy programs coming out of Keratos. So we're really excited to be launching Keratos. Keratos is the Latin word for compassion. The mission at Keratos will be to transform the lives of children and adults living with rare genetic diseases through combining advanced protein engineering and innovative vector technologies. The 3 key pillars of value for Keratos will be around programs, partnerships, and platforms. First, Keratos will have a diversified pipeline of clinical and preclinical gene therapy programs with a number of near-term value-generating milestones. Second, the entire Amicus science and discovery team under the leadership of Dr. Jill Weimer will transition to Keratos, including all the protein engineering and expertise in platform technologies. Third, in terms of partnerships, Keratos will inherit and continue the discovery partnership with the University of Pennsylvania and Jim Wilson's team, the manufacturing partnerships we have with Aldevron and Thermo Fisher, and as I just mentioned, formed a new partnership with Amicus focused on Fabre and Pompe gene therapy. Finally, and importantly, Keratos will have a very experienced rare disease leadership team. John Crowley, current CEO of Amicus, will become the CEO of Keratos, and Bradley Campbell, our current president and chief operating officer, will succeed John as CEO of Amicus. And importantly, John will remain as a chief strategic advisor for Amicus. This slide highlights the Keratos pipeline across our 3 main franchises. So first, we have our Batten disease franchise, led by clinical programs in CLN6 and CLN3, plus preclinical programs in CLN1 and other batten subtypes. Second, we have our lysosomal disease franchise, led by preclinical programs in Fabre and Pompe disease, plus we have preclinical programs in MPS IIIA and IIIB. And third, we have a broader CNS neuromuscular franchise. There, the lead programs are CDKL5 deficiency disorder and Angelman. Of note, through our collaboration, we have exclusive rights to work with the Penn gene therapy team on the majority of lysosomal storage diseases, as well as several other large diseases, including DMD, Rett, and myotonic dystrophy. So at the core of keratos discovery is really our technologies and platforms that are being developed to address some of the key challenges facing the gene therapy field today, with the focus on engineering of proteins and transgenes. So in terms of challenges around delivery, keratos has developed a number of technologies to enable and optimize cross-correction and cell and organelle targeting. Additionally, we've developed novel ways to increase the activity and potency of transgenes, which we believe will help overcome some of the safety challenges around trying to increase dose to increase efficacy. In terms of durability, keratos is also working on technologies to decrease immunogenicity of both capsids and transgene products. And on manufacturing, we have growing expertise there in analytics, including a novel in vitro potency assay platform to simultaneously assess transduction, expression, and mechanism of action in one. So this slide just highlights two of our engineering platforms that we use for lysosomal disease targets to enable and optimize cross-correction. Here, sort of in the middle to right side of that schematic, we have our platform that involves targeting transgenes and transgene products for better cell uptake and for delivery to lysosomes through using novel signal sequences for optimal secretion, but importantly also using a novel peptide fusion protein approach to allow optimal uptake through the CIMPR receptor. And we now have generated preclinical proof of concept in both Pompe and CLN1 on the ability of this targeting approach to increase the potency of transgene products. Here on the left, we have an additional platform that we now have for Fabre disease, where we use engineering of the dimer interface to engineer in disulfide bonds to increase the stability and activity of that alpha-gal enzyme. And again, we use that to optimize the cross-corrective ability of our gene therapies. As I mentioned previously, a key part of the keratos discovery engine is not only the internal protein engineering, but the collaboration with the team at Penn. And the Penn team provides a great complement to keratos, bringing deep expertise in AAV gene therapy to address those same challenges of delivery, safety, durability, and manufacturing, but coming at it from a capsid perspective versus the transgene perspective. And together we look to combine this expertise at keratos and the expertise at Penn to create potential best-in-class AAV gene therapies. So for the rest of the presentation, I will briefly highlight some of the key programs at keratos. The lead programs actually came out of work started at Nationwide Children's Hospital and Sanford on intrathecal AAV for two types of Batten disease, CLN6 and CLN3. Batten disease is a devastating neurodegenerative disease in young children. It's caused by mutations in certain CLN genes that lead to neuronal lysosomal dysfunction, and that neuronal dysfunction leads to rapid loss of motor skills, language, vision, and results in childhood mortality. We've now treated 13 children with our CLN6 gene therapy, shown here on the left-hand panel, and we've seen a slowing in the progression of the motor and language loss as measured here by the Hamburg Motor and Language Scores in treated children versus untreated children after 2 years. On the right, we have data from our CLN3 program, where we've now treated 4 children with our CLN3 gene therapy. And in that program, we've seen initial signs of stability in the progression of physical deterioration as measured here with the UBDRS physical score, again showing treated children versus untreated natural history, in this case out to 1 year. Both of these programs utilize similar capsids and transgenes, and we expect good read-through from CLN6 to CLN3. And we continue to follow these children longer term and progress our CMC and regulatory discussions to enable pivotal studies as soon as possible. The lead program in our lysosomal storage disease franchise is in Fabre disease. As described earlier, this program uses a GLA transgene engineered for greater stability and activity through creation of disulfide bonds at the dimer interface. In preclinical mouse models, as shown here in the right-hand bar chart, this engineered transgene on the far right in blue shows at similar doses, it results in much better substrate reduction versus shown here in green, the wild-type GLA transgene. And we've seen similar results here. This is kidney. We've seen similar results in the heart and in DRGs as well. Also, we've done pilot primate studies now at doses between 1 and 5 E13 per kilogram, which are sort of our target range in the clinic. And we've seen expression at those doses in primates that are analogous to the enzyme levels we see here in mice at the low and mid dose, where we see really strong substrate reduction. So we're excited to progress this program towards an IND next year. The second most advanced program in the LSD franchise is in Pompe disease. As described earlier, our Pompe gene therapy uses a GA transgene that is engineered for greater uptake through that CIMPR receptor. And again, shown here on the right, we have data from a Pompe mouse model that shows that engineered transgene in that third column, circled in green. On the top, we show muscle. On the bottom, we show spinal cord. And we see that that engineered transgene results in much better glycogen clearance than the wild-type transgene, just to the my left looking at the slide versus the engineered version. And the last program I wanted to talk about briefly is our CDKL5 deficiency program. This is our lead program in our CNS neuromuscular franchise. So CDKL5 is a very devastating pediatric seizure disorder. This is a protein that is typically not able to be secreted from cells and taken up again by neighboring cells. Here we're looking to engineer a transgene that secretes a fusion protein that includes a cell-penetrating peptide to enable that secreted CDKL5 to be taken up by neighboring cells. And we've now seen with proof of concept that we can efficiently secrete and see good cross-correction with this cell peptide fusion protein. And we hope to use platforms like this one, along with another platform we have been working on that actually involves packaging transgene products into exosomes to enable cross-correction for proteins that traditionally you can't have cross-correction, such as membrane-bound proteins. So we do think that there's real opportunity on the transgene engineering side of things to take the 5 to 20% of cells that we can transduce in a given tissue and have to hopefully turn that into better efficacy ultimately by reaching more cells through optimized cross-correction. With that, I would like to note that Keratos will be headquartered in Philadelphia, and while we are bringing over most of the key people and functions from Amicus, we will be hiring a number of new positions in the coming months. So if you have any interest, please visit the Amicus website. And in closing, I'll just note that we're really excited to continue both the mission at Amicus and also now to launch our sister company, Keratos. Thank you.